Good morning, students. We are discussing about central dogma. In that, you have seen transcription, formation of messenger RNA from DNA. Now, we will see translation, formation of protein from messenger RNA. Translation is the decoding the coded information carried by messenger RNA into protein. It's called translation, or synthesis of polypeptide chain on messenger RNA strand with the help of transfer RNA and ribosome is called translation. Translation takes place in cytoplasm. The required components for translations are messenger RNA, transfer RNA, ribosomes, amino acids, amino acyl synthetase, peptidyl synthetase, ATP, GTP, and magnesium ion. The process of translation takes place by 5 steps. First is the activation of amino acids. Second, attachment of activated amino acids to the transfer RNA. Third, initiation of polypeptide chain. Fourth, elongation of polypeptide chain. And fifth, termination of polypeptide chain. First, activation of amino acids. The specific amino acid present in cytoplasm gets activated by specific amino acyl synthetase enzyme and ATP. It forms amino acyl adenylate enzyme complex. Amino acids plus ATP plus amino acyl synthetase in presence of magnesium ion, it forms amino acyl adenylate enzyme complex plus inorganic phosphate. Attachment of activated amino acids to the transfer RNA. The DHU loop of specific transfer RNA recognizes the activated amino acid according to its anticodon. The activated amino acid binds to the 3 end of the transfer RNA. It results in the formation of amino acyl transfer RNA. See the different steps. Transfer RNA synthetase, amino acids, active sites, how the transfer RNA get attached with the 3 ends of the transfer RNA. Initiation of polypeptide chain. As in the case of prokaryotes, DNA, messenger RNA, and protein is formed. That takes place between the ribosomal subunits. Messenger RNA, AUG is the chain initiation codon, and the first amino acid, that is the methionine, that is carried by transfer RNA, UAC, that is the anticodon. Initiation by polypeptide chain. The ribosomes binds to the messenger RNA at 5 ends. The ribosome recognizes the interior initiator codon AUG on messenger RNA. The transfer RNA having anticodon UAC carries formyl methionine acid to the site of initiator codon. It initiates the polypeptide chain. Elongation of polypeptide chain. It is the linear growth of polypeptide chain on messenger RNA. The charged transfer RNA that carries a specific amino acid enters the ribosome. It attaches to the messenger RNA next to initiator codon with the help of anticodon. The peptide bond forms between these 2 amino acids by peptidyl synthetase. As the peptide bond forms, the transfer RNA becomes uncharged. It leaves the ribosome. The ribosome moves codon by codon in direction 5 to 3. As the ribosomes moves over messenger RNA, the coded information decodes and polypeptide chain into polypeptide chain. See how the messenger RNA helps in the formation of polypeptide chain. See the next, after giving that amino acids to the next transfer RNA, that transfer RNA becomes free and again ready for the charging of amino acids. And from the another end, the new transfer RNA, charged transfer RNA, comes in the ribosome and forms the peptide bond with the next amino acids. In that way, that chain of poly amino acids continues. In this diagram, see 5 amino acids are joined together and that continues. Termination of polypeptide chain. The termination of polypeptide chain takes place due to the presence of terminator codon UAA, UGA or UAG. That is also known as nonsense codon. When terminator codon comes, it does not codes for any amino acid. It leads to termination of polypeptide chain. The polypeptide chain synthesized released out from ribosome, it undergoes folding to form a specific protein. The ribosome leaves the messenger RNA. See the finally formation of that polypeptide chain. And then this polypeptide chain undergoes for modification to forms protein. Here is another diagram of this translation.